This is self-assembly of nanoscale systems using DNA-based approaches. This is a topic we started to investigate approximately 6-7 years ago. And I hope you will be able to learn what we have done during this time, and as well in the last couple of days while Emilia was busy. So before I'm going to the contents, let me just a little bit discuss the motivation. We know the progress of our society to a large degree is really due to the Industrial Revolution, which occurs in 19th and 20th century. And primarily, I would like to focus a couple of the most crucial, in my mind, criteria. One of them is ability to convert the energy from its raw to useful forms like mechanical, electrical, ability to use this energy for machine tools, new kinds of materials which became available, as well as very important from manufacturer perspective, production line. So this notion of the production lines, it's quite important because it allows not only to speed up the process, but really assemble complex systems from multiple types of components. While we have all this progress, the essential feature of this assembly or production lines kind of staying the same over, you can see in this type of example, in the last 100 years with some modification. Essentially, it's a serial approach. We have various types of components which we bring together to form some kind of devices. Even so, in Intel examples, we have to assemble, or they have to assemble CPU processor which might contain billions of transistors, and modern car has on the order of 20,000 parts. It's essentially serial approach, which required to take one by one. Maybe in some points it's possible to make it a bit more parallel. As soon as we're going into the smaller and smaller building blocks, this essential question how we build systems through the millions or billions of components become more and more important. Particularly in the contents of nanoscience, now within the last 20 years, there is a variety of nanosynthetic methods which allows to create various kinds of nanoparticles from various elements. Obviously, I don't want to emphasize this, but this is obvious, I think, today, that we can obtain new novel properties when we have not one atom and not zillions, but let's say 100 or 1,000. There are some finite size effects due to the contribution of the surfaces, due to the limited number of particles, limited number of atoms within the particles. Those effects can be both in optical area, in electrical, magnetic, and catalytic, and so on. And the question is, as soon as we have variety of those building blocks, which not necessarily only have various kinds of atomic structure, but might have also present various shapes, there is again question how we bring all this together in order to form something useful. Why we need something useful out of those materials? First of all, it's obvious, as I mentioned, that they exhibit some new properties. Another aspect, by combining various kind of components together, we can to some degree maybe mimic or create some new devices or materials. As example here, which I choose, it just is in comparison with photosynthetic process, where we have to capture sunlight, we have to be able to transfer this energy as well as convert this energy into some other form energy. All this complex biomachinery is really relies on the very precise placement of the proteins. Analogously, if you want to create devices or materials from nanoscale components, we need to find the way how we can place components in a precise manner, which is essentially give us ability, for example, in this contents to have electromagnetic field enhancement near particles, to have propagation of optical waves, or to have energy transfer between different nanoscale elements. So having this in mind, we can ask the question, do we want to use the same type of approach which require kind of one-by-one assembly, or having all these components available, we can do something better than that? And something better is pretty much brings the question, all these components are available, how we can assemble them. Some of the approaches we definitely know. And I just want to make a point, which is, again, I think it's almost obvious to make, The point is that there are many demonstrations of the system, even so, which are ranging from atoms to large-scale system, which can organize their own, create their own structures based on self-assembly. So self-assembly does not rely on this top-down, but rather than bottom-up approach, where some kind of interaction between components can lead to self-organization of those components of the system into some kind of structure. So we have examples how molecules form from atoms, how proteins can fold due to the internal interactions of the linear chains, how supramolecular structures, for example, from amphiphilic molecules can form into complex structures, and how those complex structures essentially form the membrane for the cell, which again contain thousands of different elements which coordinated in some particular structure, which also can function. Those cells form, let's say, dog, and some animals, let's say bird, can self-organize in flocks, as well as people can self-organize in society. So it's difficult to pinpoint one particular mechanism, but we understand that there is plenty of interactions. Complex interactions can lead to the self-organization process. So from this perspective, if we would like to implement this type of approach on nanoscale, essentially, what we would like to achieve through self-assembly, we would like to be able to position various elements. We would like really to have ability to create systems which contain multiple types of particles or those building blocks, because this is essentially important for functionality, as well as important in terms of technology. It's not the system which only form on its own, but we should be able to form system kind of by design rules. More than that, as you know, a cell can evolve and not be only in this particular structure. It would be great if we can form system which can be responsive, transformative, and so on. So basically, the essence of my talk is discuss those approaches which we have been developing to address this type of problems. So again, so what we're facing now, we have all these nanoscale building blocks, and we want to form some complicated object. We don't want to go through the direct one-by-one assembly. Rather than we want to rely on some self-organization of those multiple types of blocks in this particular, let's say, in some type of well-organized structure. The approach that we have been exploring, and not only we, but some other people in the field, is utilize biomolecules. So what is interesting from this perspective is biomolecules can exhibit variety of recognition properties. So basically, attaching biomolecules to nano-objects allows us to encode the way how nano-objects can interact with each other. To some degree, this is like a barcode. This particular barcode, for example, encode building 735, telephone number 631-344-3645. This is my phone number in my building where I can be found. And essentially, this allows, if we can encode those type of components, we can potentially establish the language how they can interact with each other. So this, in principle, should allow how this type of structure will be formed. What is also extremely important is reversibility of interactions. Many systems are not formed ideally from the beginning. We need to enable those type of reversibility for system to get perfected. So in terms of more specific examples, it's possible to use proteins. I will tell you more about DNA. DNA is more robust probably than proteins in terms of nanoscience attachment of the particles. Also, it's much more predictable and programmable in terms of interactions. So if we take two strands of DNA, single-stranded, which have, as you know, 4 nuclear bases, it's possible to encode, and those two strands have complementary bases, they will form double helix. This is something which is carry information for any biological object. At the same time, would like to take advantage of this very precise recognition as well as ability to program it in a relatively simple way to do the following. If we're going to attach many single-stranded DNA to the particles, and those two particles has complementary DNA, they can recognize each other and connect. As Emilia already mentioned, this type of approach has been first demonstrated by a group of Chad Mirkin and Paul Rivasades in the mid-90s. And you can ask the question, the concept is quite simple, so why it took so long really to realize this and make something useful in terms of well-defined structure? So it turns out that the difficulty comes from the following fact, that we're not only dealing with one single-stranded DNA, we're dealing with many, can be hundreds, it can be thousands, depending on the particle size. Those form shells. So the shells can have different composition, density, rigidity, and so on. All those parameters make this simple single-stranded interactions, which is pairwise in the nature, more complicated. There are plenty of collective effects. So all this needs to be taken into account. So what I will tell you more, I will tell you how we're using this type of approaches to build clusters. Clusters is probably the simplest of organization in some sense. If you consider our nanoparticle like a quasi-atom, cluster would be considered like artificial molecules. Essentially, analogously to atoms, when different types of atoms come together, like hydrogen and oxygen, they make water, which has properties not found in individual atoms. Clusters are interesting because they allow to modulate properties of individual particles. Essentially, as crystals form from atoms, superlattices or periodic structures can be formed from nanoparticles. How to do it, I will tell you. And finally, if we have ability to fabricate well-designed structures, well-organized structures, can be to some degree design on demand, I will try to tell you about our advances in this respect. So first, let me just tell you that even so those clusters seem something very simple, contain only a few particles, it's quite difficult to make them in a perfect way. The reason is that particles can interact in many various ways, and if we do it in solution, and those types of assemblies done in solution, you very quickly realize that you have various kind of multimers, and those multimers need to be purified. So this has become labor-intensive, not practical, difficult to scale. So we have developed an approach where we can eliminate many of those complications in a simple way. So we have a surface which functionalized with DNA grafted to the surface, and we have particles which has two types of DNA. One is intended for hybridization with the surface, and other for something other, which you will see right away. So what surface really does, surface allows us to block half of the particle. So when we add, let's say, another particle which has complementarity to another type of DNA, it will end up here, and this particle will be rejected by the surface. It's not going to absorb because this interaction is not recognized. More than that, when this particle will come and connect here, it will grab all available connections such that any other particle will not be able to connect. This is a quite simple approach which can remind many of you either layer-by-layer deposition or solid phase chemistry with a twist that this recognition of every new element is encoded with recognition of the previously already placed element. We can remove those simplest clusters, dimers from the surface, and we end up with this type of dimer. It's the simplest cluster which already have useful properties, which I will show you immediately. So what I wanted to emphasize here is that it's quite simple approach which allows to get 75% yield without any purification. In this particular case, we can make it from gold or from silver, and they can be useful both from optical perspective as well from biodetection perspective. But probably the most exciting is really to make structure which contain two particles, not one metallic and one semiconductor. One is gold particles, and while another one is semiconductor cadmium selenide. Why it's interesting, it turns out that if we excite, let's say, gold particles, it's a particular wavelength using laser, we can excite collective modes of electrons which give rise to something which is known as plasmon. This plasmon can interact with the fluorescence particle on quite large distances, tens of nanometers. And in particular conditions have been theoretically shown that we can have significant enhancement of fluorescence properties of these quantum dots. What is shown here are measurements using single molecular techniques, which allows basically to observed that using this type of approach and using DNA as connector, we can establish 30 nanometer separation between surfaces, and this leads to 5-time enhancement of fluorescence properties of quantum dots. I think this is the simplest structures, but this is already can motivate you to think that it's even more exciting and more interesting phenomena can be observed and translate answer to the optical study, optical applications, and so on. So while those clusters possible to make not only through self-assembly, but maybe through the lithography, there are open question how we can make three-dimensional structures. Three-dimensional structures, it's really difficult to do with anything else rather than self-assembly. So in this particular case, really the question is that how we can control the lattice of the lattice symmetry of those structures which we can form. To the first degree, can we control order at all? Or it's always a disordered aggregation, non-specific aggregation of particles. If we can, we in principle again can combine different types of particles in those superlattices analogously to atomic system. We can form system which contain either particles of different shapes, particles of different sizes, materials, and so on. I'm very glad to say that much of inspiration for this work came from theoretical work by Alex Tkachenko, who is now at CFN, who about 10 years ago wrote a theoretical paper which describes that even quite simple binary systems of two types of particles which have complementarity between each other can show quite complicated phase diagram. And the main parameters here are using the relative strengths, attraction and repulsion, and their relative lengths. So while theoretically it's very exciting for us as experimentalists, it was a question what knobs we can tune in order really to explore even the part of this phase diagram. So the approach which we have been exploring, I will tell you in a moment, relies on the following. So I discussed much this question about hybridization, which allows the recognition of the particles. At the same time, I did not tell you that this hybridization fragment, it can be not necessarily directly near the particles. It can be separated by something which we call spacer. So it looks like the spacer doesn't do any good. It only separates this recognition from the surface. It turns out this spacer is actually also single-stranded DNA, which can be quite flexible. And depending on the length of this spacer, we can tune something, we can tune softness of the repulsion. Imagine that spacer almost doesn't exist. In this case, we will be dealing with particles that pretty much have just sticky spheres. If the spacer is very long, this is a very, very floppy chain, which basically one particle will not feel in, where other particle is located. So by doing this systematic study, we try to explore how this will influence the formation of the structure. First of all, I have to mention how really we do it in practice. We have particles, and those particles with those DNAs surrounded, they need to be in solution, and we need to form, we need to probe their three-dimensional structure. So what we're using in most of the cases for three-dimensional structure, small angle X-ray scattering technique at NSLS, where we're having those aggregates in the solution with shining X-ray beam. We're collecting this two-dimensional pattern, and by analyzing the pattern, we can decide what type of structure we have. It's an extremely convenient way because it allows not only probing structure, but the probing all assembly process in situ. So as soon as we play with the spacer, we found that indeed in some particular length regime, we can obtain this beautiful pattern. I know that it maybe doesn't tell you, many of you, something, but as soon as we using this pattern, we're analyzing, we can extract something which we call structure factor, which pretty much tells you the degree of order as well as what type of order you have in a system. And this particular pattern corresponds to something we call body-centered cubic lattice. This body-centered cubic lattice is basically formed by particles which sitting in the corners of the cube, and particle which has recognition properties to those blue, red particle sits in the middle. This was the first observation of periodic structure formed by particles which interacting through the DNA. So what is important in this case, what I didn't mention explicitly, in this studies, the particle size is about 10 nanometers, and we can vary the interparticle distances depending on DNA design from somewhere around 10 nanometers to 50 nanometers or so. You very quickly can realize that these 10 nanometers particles essentially occupy a very small amount of volume in the lattice. It's only a few percent. Another maybe up to 10% DNA. The rest is really empty volume, which potentially allows many, many modifications, which I tell you later during the talk. Essentially, the particles can be generic. So in this case, it's gold, because the chemistry of attachment to gold, it's relatively simple. It's a tile chemistry. But as I will show you later, some other materials can be incorporated. So what I told you about right now, it's an approach which we call direct hybridization. That basically allow those DNA which directly attach to the particle surface to find each other and form the structure. Alternatively, we can do something a little bit different, so which we will call linker-mediated hybridization. So now particles not able to recognize each other, but this linker, which is DNA linker, can recognize something on one particle, can recognize something on another, and this will connect. Essentially, it looks like very similar. There are some practical advantage. First of all, we can have the same set of particles, and giving different types of linkers, we can generate different types of systems. More than that, our initial ideas, naive maybe by that time, was maybe just simply by giving different number of linkers per particles, we can generate different symmetries, because you would think that different types of different number, and potentially, as shown here in this two-dimensional illustration, can induce different symmetries. So this is why we study in detail this phase diagram between particles which interact through this linkers. So what we vary here, we vary how many linkers we have per particle, again, on average. And I will call them arms, as well as length. So this is R is number of arms, and L is length. The phase diagram shown here, it's not in the length, but in the units of volume fraction, which is reversely related to the length. So when the length is 0, we will be somewhere around 67%, and as soon as length is increasing, we decrease the volume fraction of particles in the solution. So it turns out that this body center cubic BCC lattice, it's very robust. No matter what, no matter how we change the length and so on, we will observe only the structure. However, the transition between, which is a good news, but however, the transition from disorder to order state, it's highly sensitive to this number of arms and their lengths. So it can be understood in the following. So as soon as we're going to the longer and longer linkers, we need more and more arms to cross these lines. This can be understood in the following scenario. When the linkers are relatively short, then one particle can feel where it's another located, and there is some incentive to go to the crystalline phase. As soon as we're going to the longer and longer DNA, there is more and more entropy within the chain, and so basically from this perspective, one particle already does not feel where another particle is located. This is something which we call entropic elasticity. And so one way to compensate it, to introduce more and more arms, which connecting different particles and overall the whole system, which gives more incentive for transition from this disorder state where they don't care about each other to the state where they do care and the crystal is formed. So from this perspective, what is also important that we found that the minimum number of connections really it's necessary to establish any crystal, it's somewhere around 8 to 10, which is corresponds to the approximately to how many particles you need to have in BCC lattice in a corner. And I know that some people can correct me in this audience to say that it's not only that, but it's also some requirement for mechanical stability of three-dimensional systems. So as soon as we're starting to use this linker, I just want to show you one of the examples that linkers can be used actually for something more than just assembly the system. We can use this more active type of linker to switch the superlattice between different type of configurations. between different lattice constants. How it can be done? I told you only about linkers which is linear and single-stranded. If we're using now, instead of linker something a little bit more complicated, I will call it DNA device, which can have two states. This is state 1, and this is basically a stem loop, which is due to some internal structure, the way this is basically we design it, or additional state that's like that. The difference between those two states, 6.5 nanometers, if we're using this DNA device now to connect a particle in a crystal, and we can switch between different states, we basically can switch the lattice constant. So the way to do it, we start from the linker, which connects to particles not shown here, and we're adding single-stranded DNA to form this type of structure, which is shown here, or remove it and going back into coil configuration, or adding additional strand to switch to this type of configuration. So in this type of approach, basically we can still preserve three-dimensional character of the system. At the same time, we can switch the system between two different lattice constants. Some of those structures, even in the relatively simple form, like body center cubic, can be already used to modulate optical properties of internal component and kind of to study the question about collective effects in three-dimensional materials, collective optic effects. So since we remember this is not just any chain, this is a DNA chain, we can encode not only ends of the chain, but also internal address in the middle of the DNA to recognize some element. The particular element that we want to use here, it's a molecular chromophore rhodamine, which recognizes its location in the middle of the DNA. So now, using this DNA linker, we can connect those two particles as well as to place in the precise position the rhodamine between in BCC in a lattice inside of the lattice. So here's an interesting question from the optical perspective. We do understand to some degree what the interactions of these plasmonic particles on fluorescence properties of this chromophore. The question is how the whole lattice will influence. Is there any collective effects, and what are they? So in order to probe this, we can do even something more than that. We can change the lattice constant of this crystal in the continuous manner. We just have to remember that DNA has negatively charged backbone, and simply by changing the salt concentration, we're changing the persistence length. This is degree of rigidity of DNA, which allows to this whole crystal to be swollen and contracted. We can do it in situ. At the same time, we can measure using small angle scattering all the structural requirements, and we can do optical studies on this type of system, basically to study what the change of the fluorescence lifetime of this rhodamine while we're doing all this modification. I will show you here the summary of those results, which is shown here as the following. This is the distance between this rhodamine and the closest neighbor, gold particles, and what is shown here, it's a relative change of lifetime. You can see that we can vary approximately by factor 2 from about 10 to about 19 nanometers the distance, and this effect in nearly 3 times change in a lifetime of rhodamine. More than that, we can model this type of systems in more details, take into account how many gold particles surrounding and what distances they located, and from this we can quantitatively understand this relationship. So what we found out that really this change of the fluorescence lifetime of chromophore, it's really sensitive to the large degree only to the first 2 coordination spheres. What is important, that number of neighbors and their location pretty much determines the optical response. At the same time, it's clear from this type of experiment that maybe precise 3-dimensional structure over long range order, it's not necessarily important here, but a short range neighbors position is really important for optical response of rhodamine. What I told you so far, it only was about spherical particles, which by itself quite complicated objects if you just think from the perspective that really the strictly mathematically maximum packing for spherical objects have been only proven about 10 years ago. And you can imagine with all the variety of solid shapes which are possible to form, there are many purely mathematical questions as well as the questions related to the packing of those objects which need to take into account entropy effects and so on. The points, however, which I want to make, it's beyond solid objects. What is special about nanoscale? What is special about nanoscale? That variety of shapes which we can create today from atoms on nanoscales, as done, or most of the time, are done through wet chemistry. So we're using some type of ligands. In any of those cases, we cannot talk only about solid objects because there is always effect of ligands. Those ligands, to a large degree, can mask the shape. They can provide some directional interactions. There are some collective effects associated with those chains. So the question becomes even more complicated. So it's not only about packing of solid objects. It's a pack of interplay chains and shapes. So I will give you just one example, which demonstrate that indeed chains can have some profound effect on organization of objects. And this is an example with rods. Rods, it's something which is very, very basic, and many of the organization rods are not really related to the interactions, but rather related purely to the geometric effect. This is something which have been extensively studied in the middle of the 20th century by Anzager, who understood that really one of the major factors in most of those systems is volume fraction, which can lead to formation of different phases known as nematic, smectic, or various kinds of smectic. What is also known in the last 10 years, there are plenty of nanoscale systems which demonstrate this effect. By the way, this effect is so common that it's found for molecules, for viruses, and if one wants to do experiments with pencil, you probably can obtain this with pencil as well. But for nanoscale objects, there is typically some kind of interactions. This type of interactions between objects can allow for some kind of registry, and in many cases, smectic phases or something which is known as smectic B phase is observed, where a particle can form in-plane hexagonal type of structure. So as soon as we're dealing with nanoscale objects, rods, and they functionalized with DNA, we're not dealing with particle which of only the same type. We can deal with a particle which have two types of determined by those DNA recognition and DNA functionalization of rods. So we very quickly can realize the following. The hexagonal structure, it's not necessarily going to satisfy to those interactions. A doesn't like A, B doesn't like B, but A likes B. So this is difficult to satisfy. There is a frustration. This guy not clear what it's supposed to be, A and B. From this perspective, square lattice seems to be something which should satisfy this condition. This is something which we kind of expected starting this type of experiment. Nevertheless, we observed something completely different. And what it is, it turns out they don't form any membrane, two-dimensional type of structure. Rather, they're forming one-dimensional ribbon type of structures. What you see here is electron microscopy from different types of rods which functionalized with different lengths of DNA. As you can see immediately, there is different inter-rod distances, and there is different degree of flexibility within the ribbon, but they do form these ribbon structures. This can be understood not only from the perspective of rods, but it's really necessary to take into account that there is interplay of shapes as well as all those chains which connected to the rod. So this is what is shown here. It's the side view of rod, and they have those many chains attached. If chains are long enough and flexible enough, when two rods come together and chains want to hybridize, they will be able to be consumed within those two cylinders between the rods. So when another particle will come and it's supposed it has the right recognition, it will not be able to join anywhere here. It will basically be forced to join in linear fashions which promote linear growth, something which you have seen here on the top. We can do much more detailed study of the kinetic of this evolution using X-ray scattering. Again, small angle X-ray scattering provide time dependence and whole details of evolution. So we can start. We can see immediately in the initial stages of this type of structure factor, which corresponds to the formation of these ribbons, which I mentioned. However, at later stages, we see the development of additional peak, which can be associated with ribbon stacking. So if you remember that DNA allows us for reversibility of this interaction, so those ribbons start to interact and exchange the DNAs between those rods, which lead to the weak binding between rods and this type of stacking. So from this point, I just want again to emphasize those chains allows really they enrich significantly the behavior, and this is something which need to be understood better. So what I would like to emphasize now is conceptually it seems quite simple. We can take various kinds of particles and bring them together using this DNA recognition. More than that, we can adjust interparticle distances through DNA design, and as I showed you previously, it can be even switchable. And so this seems to be so simple that we can ask the question why we don't take all these particles and will be enabled to create a variety of materials, and it seems like this should be possible. Why it's interesting, if we can bring different types of particles, we not only making heterogeneous structures, we can make materials which are really truly multifunctional because those particles can carry different type of functionality. While it's so appealing, the difficulty, however, is different sizes of particles can affect different packing. There is different chemistry of attachment which need to be realized for those type of particles. There are different shapes, and all this become a bit complications which are necessary to overcome. So what I would like to show you example of the system where we have been able to overcome and build three-dimensional superlattice which contain both gold particles, which is plasmonic elements, and fluorescence particles, which are quantum dots, cadmium selenide particles, which is also, apart from this perspective of functionality potential, it also allows to ask interesting question. I showed you before this type of structure, and I told you this is gold particles, this gold, and the color, it's only due to different DNA sequences. However, you can ask me how I know about something like this, because essentially for X-ray, they have the same color. It's only in terms of recognition. To address this question and to know truly, do we have a well-ordered structure where there is no compositional disorder? That means that we not only have one-to-one ratio on average, but we do have indeed the location of those particles in the middle of this cube. It's necessary to have some kind of different color. The different color for X-ray is different electron density. So this is interesting question, and so this is possible to do. For example, if we're taking quantum dot, and this quantum dot has not only, it also has polymer shell around. This polymer shell has two functions. First of all, it allows to do the chemistry not on the surface of quantum dot, but on a polymer shell, which preserve completely optical properties of those quantum dots. As a second, we can match the size that the quantum dot with the shell has exactly the same size as the gold particle which we use as in a control experiment. As you can see here, you cannot distinguish. So basically, we're truly substituting here the gold particle with this quantum dot. If we measure these two types of systems, and because we know all distances, as I mentioned, we know everything about individual particles, we can analyze the scattering from both types of systems and compare. I'm not going to go into details, but basically we can conclude that this type of structure, in this type of structure, indeed quantum gold is located in the middle of this cube, and we can form three-dimensional body-center cubic structure from two types of particles. And there are many interesting optical effects which I'm not going to tell you today about, but it's essentially we can manipulate the fluorescence properties of quantum dot using this three-dimensional assembly. What is more, it's not, those particles doesn't have to be quantum dots. Recently, we have been able to basically to demonstrate that we can use basically plasmonic particles as a gold magnetic particles as iron oxide, catalytic particles, for example, as palladium, of various shapes, octahedra, cubes, and rhombic shape, as well as fluorescence particles, both cadmium selenide and cadmium telluride. And all those materials can pretty much mix and match in order to form this three-dimensional organization using either direct hybridization or through the linkers, all those approaches which I mentioned previously. So what this allows is really allows for in terms of materials to make multifunctional materials which can be at the same time fluorescence and catalytic or magnetic and plasmonic and so on. What I show you here, the phase, it's not really the phase diagram, but rather a library of materials this from binary components which we can assemble, and those symbols represent what type of structures we can observe in those materials. We can see that we are in the body center cubic BCC lattice, it's quite robust, and at the same time we can observe something which is more like face center cubic system as well as the clusters. More detailed information reveal that on those data systems reveals that we can also understand to some degree where the system goes from the clustering behavior to the crystalline. For particles of similar sizes, one of the major parameters is the number of linkers or number of arms which particle, DNA arms which particle has, and when it's around 20 or 30, typically clustering occurs. More than that, typically we can have transition to the crystalline organization of those materials. So from this perspective, I would like to spend some time telling you about remaining challenges, and so what really exciting work can be done, and I will show you some of the examples along those lines. So really, the question, we can form some relatively simple symmetries from particles, and the question would be, if you really would like to design symmetries and form more complicated lattices and so on, how we can do something like this? Again, taking into account this as a complicated question, I will address this in more details. I would like to mention there are some other interesting questions. We now have ability to form crystalline organization. At the same time, it would be interesting to ask the question, can we truly through self-assembly induce some morphological formation of large-scale structures like, diamond, it's only valuable where it has particular appearance in its macroscopic shape, not necessarily only if, in a crystalline shape. Another question is, can we find the way to use self-assembly to form not only periodic but non-periodic arrays or non-periodic structures? The questions about switchable system very quickly bring to the question about a system with which behavior not necessarily static but can develop with the time. Those systems might consume some kind of energy and be able to develop and or evolve in some way. How it's possible to do? Another question, as you understand, many of those systems not necessarily form in the perfect way. What would be the way to have some automatic error correction or healing of the system? Pretty much, I want not only to tease your imagination, but I want to say this is the type of questions that we are concerned with, and this is something which we are working on. But let me just focus on one particular example and asking the question, how we can design the symmetries of those three-dimensional systems. So I told you before about recognition. Recognition between particles is quite powerful, but is it enough? It turns out that in order to form quite complicated structure, it's not enough really to establish the interaction between neighbors. It's necessary to some degree prescribe how next neighbor will be placed and so on. So this kind of easy instruction, which I have been trying to tell you about using DNA, not so easy to do if you think from this perspective. How it's possible to do? Computational work, for example, shows that if you have isotropic interaction between particles, and if you can shape this isotropic interaction in some particular form, you can induce various kind of organization. For example, this potential lead to honeycomb, and more complicated potential with this position of the particles, where they need to be can lead to diamond structure. So this is interesting from computational perspective, but from practical perspective, it's very difficult really to engineer interparticle potential. Alternative approach can be thought if we're looking at atoms. Atoms typically have bonds which are very well defined and has particular geometry, and this is why, for example, carbon with bonds organized in this tetrahedral form can form diamond lattice. So how it's possible to use this for particles? Again, computational work for in the last 20 years suggested that if you can have patchy particles, those particles which have particular binding patches, so those patches basically just provide attractive points for other particles, and this, if we can place them in the particular position of the particle, they can promote formation of different structures. This is something which might be possible to implement for micron-sized particles, but become extremely difficult to implement on nanoscale, just because it's simply very difficult to control where is your patch located, and as you can imagine, the symmetry of the whole structure will be strongly influenced all kinds of imperfections. So it's not really practical on nanoscale. So we're facing this problem that how we can engineer those three-dimensional structures. From one side, it's difficult to engineer isotropic potential. From the other side, it's difficult to fabricate those patchy particles. So the idea is to use linkers which have some symmetry of valence bond. What does it mean? All those examples that I showed you before, we take into account some conventional linker, which is basically some kind of chains which are able to connect particles. So what if linker has some kind of symmetry? So in this case, if the linker has some kind of symmetry, and again, it has the right size in comparison with the particle, it can organize particle in some particular clusters. This will promote formation of, this geometrical effect will promote formation of larger scale structures. So what is important, we're not giving up DNA. DNA is extremely important for recognition of the local recognition here. However, the long range order is promoted to some degree these geometrical constraints imposed by those linkers. So a little bit more practical, it's possible to use more complex DNA constructs, which I'm not going to discuss in detail as for example, or it's possible to use anisotropic particles as those types of linkers. And this is as shown here. One can imagine we can use more complex organization of spheres, for example, promoting their connection with cubes. In the last couple of minutes, I just want to show you this is not just a dream and not just an idea, but it's possible to realize. And so in this case, we're using cubes and we're using spheres. As I showed in the previous slide, we're trying to achieve this type of organization from clusters to lattices, and this type of lattice, basically, it's a sodium chloride of lattice. Indeed, we can observe using electron microscopy, as well as small angle scattering, which is not shown here, that we can start from forming small clusters, and those clusters eventually form large-scale structures which contain alternating cubes and spheres in this type of manner, pretty much according to our prescription. Even more complicated structures can be promoted if we use different other geometrical objects. For example, we're using, in this case, octahedra. And using octahedra, one can imagine that it's possible to induce a structure, which is a cubic structure, which is known as perovskites. This is a very common structure for many people, I guess, in material science and condensed matter physics department here, because many of those oxides exhibit very interesting magnetic and electronic properties. So we can pretty much replicate this type of structure on nanoscale, again, by design, inducing this formation using this octahedra. I mentioned something about DNA construct, so I just want to give you an example. We can use now tetrahedra. In this case, it's not a nanoscale object which is inorganic, rather it's a DNA construct which contains DNA which able to organize in this tetrahedra type of organization. They have 4 arms. I mean, which have different recognition. In this case, they have exactly the same recognition to the particle. If we're using this as a connector between the particles, we can promote the structure, which is a diamond type of structure, as can be confirmed by our small angle scattering measurements. So this is quite unique structure. It's very difficult to obtain diamond, and so this is something which we are very excited about. So finalizing, I just want again, there are many other interesting aspects which I already mentioned, which has to do with control of macroscopic organization, non-periodic arrays, regulated dynamical behavior, error correction, and of course exploration of all those interesting optical and magnetic and other properties which can be induced by bringing different types of particles together. So within this, I would like to summarize, and basically I have been trying to convince you that DNA is really a great tool to control and regulate nanoparticle assembly. We can use this for forming both clusters, three-dimensional lattices, as well as structures which has, let's say, some degree of functionality, like optical functionality. I think that it shows potential for future structures. What is more important also is that those structures not necessarily have to be static. The structures can be switched on demand, as I showed you in three-dimensional assembly. There are many interesting effects associated, which we need to understand, associated with interplay of shapes as well as chains on nanoscale, and with all this diversity of structures which we'll be able now to assemble. I think there is a great potential for material science to explore new materials. So finally, I really would like to say this is not my work. This is the work of many, many people with whom I was fortunate to collaborate on this project. I would like to name all of them because they all play a very important role in this type of study. Dmitro, who is, I hope, somewhere here, was the first postdoc, and now he is with me when we started this in 2004, 2005. So he's now at CFN. Matthew May, who is now professor at Syracuse, was postdoc with me. Hui Min, who is now professor at Shanghai. Daniel van der Lille, who was a collaborator, close collaborator, who is now at RTI. Alex Tkachenko, who is a theoretician with whom great to have it here, not remotely at Michigan as before, but on site. Bill Sherman and Kumara, who helped a lot with the switchable structures. Matt Fire and Mircea Kotlet, who are great resource for many optical questions and measurements which we can conduct at CFN. Cheng Chi, who is a student from Stony Brook, has been working actively on clusters and the application for detection. Stephanie Vial, visiting scientist now in Portugal. Peter Sand, who has been working a lot on quantum dot and plasmonic type of systems. Andrea Stadler, who now at St. Joseph University professor, who has been working on biodetection aspects. Fan Glue has been working on those beautiful perovskite type of structures and cubes-induced assembly. Yu Gang Zhang, who has been working on heterogeneous structures and all this diversity of mix and match type of approaches. Miha, who is Tagawa from Japan, who is visiting scientist and who has been able to obtain this diamond type of structure which I showed, and as well as Kevin Jager, who helps a lot in various small angle scattering analysis, and Sunita, who has been working on particle and two-dimensional surfaces. As well as there are many, many colleagues at NSLS, at condensed matter physics and biology department, which I would like to acknowledge, as well as our facilities at CFN, which doesn't have to be introduced additionally at Brookhaven, as well as many of our measurements were conducted at different beam lines at NSLS. And obviously, I would like to acknowledge the support from DOE. Thank you.
Thank you very much, Oleg. What is the thermostability of these structures?
The thermal stability is determined pretty much the hybridization and dehybridization of DNA. Most of the cases, it's somewhere from room temperature to 60, 70 degrees Celsius. So that's pretty much what DNA allows to do.
So I was curious about the space between the nanoparticles. Tries placing closely at DNA, and there are many biological materials that bind that DNA and distort it in known ways. Have you exploited this as a way to give you some extra topological flexibility?
It's a good question, and thank you for asking, because I definitely did not show everything that I have, and this is a good opportunity to say something about this. Yes, indeed, it's possible to use various type of interactions as a protein-DNA interaction, so site-specific intercalators to induce either changes or to do something more interesting. So, for example, we have been using PNA. It's an analog of DNA which doesn't have charge, to induce this site-specific intercalation. And this is one of the routes which we're exploring for arbitrary, kind of by design, assembly, non-periodic structures.
For a particular application you're directing
Could you be a little bit more specific? Honestly, I will tell you that we are very much interested in exploring principles and figuring this out and revealing. At the same time, I wanted to include, and I did include, a couple of, I would not call them application, but I would rather say demonstration that we can change optical activity of underlying elements when we're building this type of structures. So from this perspective, the next question about application has to do, yes, we can do applications. We have a couple of patents in biodetection. But in terms of materials, many of those principles, as soon as we figure out, can be transferred to materials which not necessarily based on DNA. But there is a question that you need to have some kind of understanding with the model system. DNA does provide this. So another answer would be to this question. I know this is something which many people have been thinking probably through the talk. What is this good for, right? And I would like to say that in many cases, the performance of solar cells or in many other energy converting or energy transferring devices relates on the placement and precise placement of antenna, fluorescent center, and so on. We have to figure out how to do it. This is one of the approaches, how to do it. And we need to understand if we can figure out how to do it, how it really works. This is, again, one of the approaches which allows us to explore and understand this.
for spacing DNA, the non-hybridizing DNA, just to try to control the placement of particles. Do you look at various compatibility properties of binding peptides? You make these sequences. Do you worry about that?
Typically, we're trying to avoid this. For us, it's more like a parasitic effect, so we're trying to have it as random as possible to minimize it. But again, it can be used to advantage. And so in this case, where we have a switchable device where it's forming stem loop, this was used to advantage to stabilize the stem loop and to form some secondary structure within this. So it can be, depends on what we need to achieve, we can use both. Yeah.
So can you modify any of these structures or lattice parameters by modifying the
Yeah, definitely. There is a great possibility to modify it. I mentioned two of those examples. One of them through switching, another through changing the salt concentration. And this allows to modify, depending on salt and so on, probably by factor 2 or so. So that's possible to do. There are other factors like pH and so on, which again can promote a variety of switching and so on by incorporation of different motifs. realistically, I don't think that those structures are really intended to withstand any elevated temperature. And this is what I want to emphasize. They live in a kind of environment maybe up to 80 or 90 degrees Celsius with current design. What I would like to emphasize, and I thank you for this question, the same way that we can look at the polymers widely used in nanofabrication as a materials to fabricate something and later get rid of it, this type of approach can be used for form structure. Later, we can think about making various kind of modification, chemical modification, to freeze the structure. And when it's solidified, we can get rid of the DNA, or basically DNA can be mineralized, metallized, or whatever it is. And it's only used as really a driving force for assembly. So I think from this perspective, rather than that those structures necessarily directly used for applications.